it is my distinct honor and privilege to introduce today to you Dr. Srinivas Sharamula, and he is a newly tenured associate professor, congratulations, in the Department of Pharmacology and Toxicology at East Carolina University. He is been educated and a degree in veterinary science from ANGR Agricultural University in India, and has a PhD in veterinary medicine from Louisiana State University, Louisiana. The major goal of his laboratory research is to elucidate mechanisms underlying the pathophysiology and identification of novel potential therapeutic targets for the treatment of cardiovascular and neurodegenerative diseases, including Alzheimer's disease. He is examining neuroinflammation in neurodegenerative disorders and studying the SARS CoV-2 infection in a mouse model to investigate long-term consequences of COVID-19 on inflammation, cognition, neuropsychiatric symptoms, and the possibility of increased risk of developing neurodegenerative diseases such as Parkinson's disease and Alzheimer's disease. I will turn it over now to Srinivas, and we're looking forward to your talk.
Thank you, Donna, for that nice, generous introduction. And thank you, Duke-UNC Ideas Forum, for giving me this opportunity to share my research. So let me share. Okay. I hope everyone can see my screen. Thank you. So since I was a graduate student, I was focused on understanding the role of inflammation in cardiovascular diseases, and now I'm expanding that into neurodegenerative diseases. So title of my talk today, I wanted to focus on the new direction of my lab's research, understanding the COVID-19 pathology, whether Kinin B1 receptor can be a therapeutic target. So today I would like to give a brief background, and then I'll go over the kallikrein kinin system, which is focus of my lab, and then how we wanted to develop and characterize the mouse model to study the persistent effects of the SARS CoV-2 in long term, and then how the neuroinflammation and the Kinin B1 receptor plays a role in COVID-19 pathophysiology. And then I wanted to focus on some ongoing research and unanswered questions, which are very much important to answer. Just a brief background. It is now well established that the neuroimmune interactions or the communication between the brain and immune system are an important part of health as well as the disease states. So the brain sends signals to the immune system through neuroendocrine pathways, including the hypothalamic pituitary adrenal axis, and then through the autonomic nervous system, specifically in the cardiovascular diseases. Then the immune system interacts with the brain through the elevated pro-inflammatory cytokines, as well as disrupting the blood-brain barrier. So these elevated cytokines, as well as the disruption of blood-brain barrier, results in the neuroinflammation, heightened neuroinflammation in the brain, which is mainly partly through the activated microglia. So this heightened state of neuroinflammation is mainly through the intercellular feedback loops between the reactive microglia, astroglia, and the neurons. So under the normal conditions, the quiescent microglia can become reactive microglia and then secrete pro-inflammatory cytokines. While doing this increased inflammatory status, then it can receive signals, or it can send signals through the astroglia in terms of like astrogliosis, or it can receive signals from the neurons, and it can also signal the neurons. So understanding these communication loops between the various cell population is important to understand the disease status as well as under normal physiology. So my interest in studying this inflammatory role in the brain in outcomes of the cardiovascular diseases started when I was a graduate student at Louisiana State University School of Veterinary Medicine under Dr. Joseph Francis's lab. So as part of my dissertation project, I was more focused on an hypertension model and understanding how the inflammation in the brain increases sympathetic activity and then hypertension. So I was able to show using a mouse model of hypertension as well as a knockout for inflammatory cytokines such as tumor necrosis factor alpha. The inflammation is indeed involved in the pathophysiology of hypertension. I was able to also show that the TNF inhibition using a whole body knockout mouse model or using a specific antagonist against TNF alpha. That if you block TNF alpha, then it can attenuate
Yeah, I got.
and end organ damage. So, as I mentioned, the microglia plays an important role in the neuroinflammation. We were able to show that the brain microglia are the major source of cytokine production during the neurogenic hypertension. And then we identified NF-kappa B as an important transcription factor in excited neurohumoral excitation during hypertension as well as heart failure models. So these studies, my PhD studies were clearly showed evidence that the inflammation within the brain cardiovascular regulator centers can play a major role in hypertension. Then I extended these studies when I joined as a postdoctoral fellow in Dr. Eric Lazartik's lab in the LSU Health Sciences Center, New Orleans. Then we focused on angiotensin converting enzyme because the component, this angiotensin converting enzyme 2, now everyone knows about this because it's a receptor for the SARS CoV-2 virus. ACE2 is part of the renin-angiotensin system, which is an important system for the cardiovascular diseases, especially hypertension and heart failure, plays a role in mediating inflammation and sympathetic activity. So Dr. Eric Lazartik is a pioneer in ACE2-mediated signaling mechanisms. So in his lab, I was able to show that the ACE2 overexpression in the brain can benefit in preventing the development of hypertension. And we also found out that the ADAM17 TACE, TNF alpha converting enzyme, which is responsible for shedding or cleaving of ACE2 in the brain, that ultimately leads to hypertension. So during my postdoctoral tenure, I wanted to develop my own project. So I was looking for some targets. So one of the close system, that is the kallikrein kinase system that interacts with the renin-angiotensin system. So I thought that maybe since the bradykinin can be cleaved by angiotensin converting enzyme into inactive metabolites, as well as the bradykinin product, the disarginine bradykinin can be inactivated by angiotensin converting enzyme 2 into inactive metabolites, suggesting that there is a crosstalk between these two systems. Since ACE2 plays a role in hypertension, I was maybe proposing that kinins might have some role in hypertension. So I was started some literature search and collecting some preliminary data. And I was applying for the American Heart Association postdoctoral fellowship to develop this line of research. So why kinins? Why kinins? So just to give a brief background, it dated back to the early 1900s. So the first time in 1925, Dr. Emil Carl Frey identified that if we inject human urine into dogs, then it can lead to decrease in blood pressure. Later, it was isolated and characterized as an hypertensive agent named as kallikrein. However, after 10 years, it was realized that this is not the kallikrein, but the kallikrein converts a precursor in the blood into a bradykinin, which is actually hypertensive agent. So in 1949, Dr. Rocha Silva isolated the bradykinin from the snake venom. And by 1980, they were able to identify the structure of both the two G protein coupled receptors that mediates the functional aspects of the kallikrein kinase system. Even though these receptors are identified in 1980, it took another 20 years to develop knockout mouse models so that we can fully understand their physiological as well as pathophysiological roles. Once these technological advances were made, within 8 years, there was a first drug, a B2 receptor antagonist, icatibant, was FDA approved for the treatment of hereditary angioedema. However, still there is a lot much not known about the B1 receptor, and that's where my research direction was developed. So I was hypothesizing that it may be play a role in neurogenic hypertension at that time, and now it's in COVID-19. So what is this B1 receptor? So if you look at these precursors of the kininogens, high molecular weight kininogen in case of plasma, low molecular weight kininogen in case of tissue, these are activated by plasma. These are cleared by plasma kallikrein or tissue kallikrein into active metabolites. So the bradykinin, which is a 9 amino acid peptide, and the kallidin, or 10 amino acid peptide. So these bradykinin and kallidin can act through the B2 receptors. However, when an enzyme kinase 1 can cleave one amino acid from this carboxy terminal, then it leads to an entirely different endogenous agonist. So it forms from the bradykinin, desRG9 bradykinin, from the kallidin, desRG10 kallidin. Just one amino acid difference between these two. This one amino acid can change their specificity towards a completely different receptor, that is the bradykinin 1 receptor. If you look at these functions of these two receptors, the bradykinin or kinin B2 receptor is mainly constitutively expressed everywhere, and activation of this receptor by endogenous ligands can lead to vasodilation via nitric oxide production. On the other hand, the kinin B1 receptor is not constitutively expressed, but it is inducible either at the sites of injury or through the inflammation. Once this B1 receptor is activated by the endogenous ligands, or it can be also activated by inflammatory cytokines or even the lipopolysaccharide. So this leads to either vasoconstriction or further increasing the inflammation or immune cell infiltration. This role in inflammation was the basis that I was proposing that the kinin B1 receptor maybe is involved in hypertension or any neuroinflammation mediated diseases, including the neurodegenerative diseases. So in 2017, I moved to ECU along with my postdoctoral fellowship as well as the direction of the kinins. So I continued to explore the role of B1 receptor in neuroinflammation further. So I was focusing on mouse primary hypothalamic neurons, and I was trying to understand the relationship between the renin-angiotensin system. The main effector peptide of the renin-angiotensin system is the angiotensin 2, which acts through the AT1 receptor as well as the B1 receptor. So our initial study showed that the angiotensin 2 can activate AT1 receptor, which in turn cross-reacts with the B1 receptor in leading to increase in oxidative stress and neuroinflammation. And when we block this B1 receptor activation via a peptide antagonist against B1 receptor or 715, it can mitigate neuroinflammation and oxidative stress. We extended these studies in vivo using B1 receptor whole body knockout mouse or receptor antagonist, finally showing that there is a nice pathway going on. Like, for example, the B1 receptor can be activated during the hypertension, which is due to increasing chronic inflammation, oxidative stress, and increased sympathetic activity. But the important thing about the B1 receptor is that once it is activated, it can cause a vicious cycle of a positive feedback loop in further increasing the inflammation. Not only that, this receptor can interact with other receptors such as angiotensin 2 type 1 receptor or through the ADAM17. So these ADAM17 and AT1R can increase the ACE2 shedding. So if you reduce the ACE2 levels, actually it is shown to be causing hypertension. So that means if you restore the ACE2 levels, it can actually mitigate the hypertensive phenotype. So these studies were the clearly basis that the B1 receptor plays a major role in neuroinflammation. So during that time, COVID-19 pandemic hit. So although COVID-19 is no longer defined as a public health emergency, however, it continues to have a significant toll on health globally. As you can see, it first started in December 2019, but in the last 4 years, still we don't know much about the sequelae of this COVID-19. So initially, the acute COVID-19 has devastating effects on increased mortality. However, now with the advent of the COVID-19 vaccines as well as treatment options, now we are more focusing on the long COVID, so the multi-organ systems after COVID-19 infection. In order to understand the differences between acute and the long COVID, so just to briefly go over the acute COVID-19. So SARS-CoV-2 binds with the ACE2 receptor in the epithelial cells, the respiratory system, through the respiratory system. Then immediately it causes an autoimmune activation within the respiratory system that leads to microclot formation and then the cytokine storm. So this initial cytokine storm can trigger further chronic inflammation, and then this leads to the multi-organ damage, complement activation, clot formation, cytokine and bradykinin storm that is associated with increasing inflammatory state throughout the body. So this acute reaction can lead to multiple dysregulations, including immunological, neurological, as well as the pulmonary dysregulation. However, how this acute infection can lead to long COVID symptoms. So one of the important expectations or suggestions is that SARS-CoV-2 can have either direct neuroinvasion effects, or it's indirectly through the cytokine storm. So neuroinvasion potential means the virus, the SARS-CoV-2 virus, can reach the brain through the circulation, either through attaching to the endothelial cells, which expresses ACE2, through the immune cells. It can reach the blood, it can reach the brain, or it can travel through the retrograde transport through the axons of the nerves, such as vagus nerve and olfactory nerve. And also peripherally, it can increase the cytokine storm that can cause increased blood-brain barrier permeability, which can lead to increased neuroinflammation. So within the neurons expresses ACE2 receptor. That means a SARS-CoV-2 virus can enter into the neurons. Still, there is a debate going on whether the SARS-CoV-2 mediated effects are due to direct neuroinvasion or due to indirect effects through some other signaling mechanisms. However, evidence has been shown that it can cause lysosomal, mitochondrial, or endoplasmic reticulum disturbances, and it can lead to protein aggregates and can cause neuronal damage. So these effects can lead to the long COVID or the post-acute sequelae of SARS-CoV-2 infection. So these are like symptoms that persist beyond the initial phase of the SARS-CoV-2 infection, and then it is defined by WHO that it can last up to 3 months after the initial SARS-CoV-2 infection and can last up to 2 months with no other explanation. It is expected that up to 30% of patients with COVID-19 experience some persistent symptoms after recovery from COVID-19. And approximately 1 in 5 adults have some illnesses such as neurological or mental health conditions, renal failure, musculoskeletal conditions, cardiovascular, respiratory, or blood clots. Given the pandemic levels, like more than 500 million people around the world had COVID, that it means that there is possibility that millions of people could have some type of long-term health effects. So understanding this long-term health effects of recovery from SARS-CoV-2 infection is very much essential. So during that time, I was interested in understanding, studying the SARS-CoV-2 effects. So there are several animal models to study COVID-19. Some are very good for the acute COVID. Some suggested that it can be useful models for the long COVID models. However, I was looking at the options. So during that time, my friend and colleague, Dr. Eels, he was an expert in understanding the neurodegeneration and the dopamine neurons in Parkinson's disease. So he was working with K18 human H2 mice, and he was asking me, "You want to work with COVID-19?" I said, "Why not? I'm also interested in understanding the neuroinflammation and its potential in causing the COVID-19 syndrome." So I was at the same time, there are several speculations or speculated review articles. There is an exponential increase in the articles related to COVID-19 during that period. So I was looking at some opinion articles, some evidence-based link that the kallikrein kinase system as well as the renin-angiotensin system plays a role in COVID-19 pathophysiology or pathology or outcomes. So if you look at the bradykinin B1 receptor, which is part of the kallikrein kinase system, and then the ACE2, which is the part of the renin-angiotensin system, both are involved in um, COVID-19 infection. At the same time, there is evidence that the bradykinin B1 receptor knockout mouse will have some positive outcomes in cognitive and cerebrovascular improvements. And also there is a B1 receptor antagonist has shown that it has some beneficial effects in Alzheimer's disease model, which is APP mice. So that means the B1 receptor can have some role in neurodegenerative diseases. So with that, since the SARS CoV-2 has some evidence for some psychiatric disorders which are part of the long COVID, we wanted to have explore further the role of B1 receptor in SARS-CoV-2 mediated neuroinflammation as well as psychiatric or behavioral changes. So with that background, we hypothesized that the SARS-CoV-2 infection induces long-lasting B1 receptor expression in the brain, driving chronic inflammation, then ultimately causing cognitive and neuropsychiatric symptoms. So we first wanted to establish that the B1 receptor is indeed activated during the SARS-CoV infection, both in acute and chronic cases. And then we wanted to look at the hallmarks of COVID-19, that is lung fibrosis and neuroinflammation and neurocognitive changes. First, we wanted to establish a model to study the long COVID. So at that time, we used the K18 human H2 model, which is usually the virus infection of the K18 human H2 induces the severe infection. So we wanted to titrate the virus dose so that the mice can survive. So most of the mice with SARS-CoV-2 infection, K18 human H2 were died within 10 days post-infection. So we wanted to titrate the virus dose so that the mice can survive further. So we used low dose, intermediate dose, and then high dose. Then looked at the body weight loss as well as the survival probability. As you can see, we did not see any significant differences in these three. So these three, even though we use three doses, these three doses are somewhat lower than some doses used in other severe COVID-19 or severe acute COVID-19 disease models. So there is no significant difference between all these three viral infection groups. However, when we see the mortality, in case of the intermediate dose, there was like a 20% of mice died by days 9. In case of the higher dose, the 67% mice died. So we don't want 100% mice died because we just wanted to study how well we can prolong the survival of these mice. So we chose this high dose as a acute severe COVID-19 disease model, and then we chose this intermediate dose as a mild disease or mild to moderate form of disease. So we started with the acute severe SARS-CoV-2 infection. So we infected the mice with a higher dose, and then we wanted to see the neurotropism of this virus or cell tropism of this virus. So using the antibody against the SARS-CoV spike S1 protein, so we were able to show that there is an increased virus presence in the lungs of these high dose infected mice. Then importantly, the olfactory bulb on the brain, so SARS-CoV-2 virus was present. So we used two different antibodies just to be sure. One is the spike protein, the other one is the SARS-CoV virus antibody developed by BEI. Then we also used an antibody against double-stranded RNA, which showed the presence of virus in the SARS-CoV-2 infected mice. So this antibody against dsRNA indicates active viral replication. That means not only the spike protein or just the virus is present in the brain, but it is actively replicating within the brain during the early 10 days of acute SARS-CoV-2 infection. Then we wanted to look at the inflammatory markers. As you can see, it was speculated that the SARS-CoV-2 can activate angiotensin 81 receptor, TNF-alpha, IL-6, and then this cytokine storm can lead to acute respiratory distress syndrome, multi-organ failure, or coagulation deficiencies. So we started with IL-6 and TNF-alpha. We are focusing on three important regions like amygdala, prefrontal cortex, and hippocampus. As you can see, compared to the mock-treated control mice in the SARS-CoV-2 infected mice, there is an increased expression. Here is the quantifying data. There is a significant increase in the IL-6 immunoreactivity and TNF-alpha expression in the amygdala region. I just wanted to point out one thing is that all these studies were performed in BSL-3 facilities, and we were able to get only the tissues which were stored in formalin for long periods of time. Then only we were able to get those tissues out. So that limited our capacity to perform certain experiments. So we mostly relied on whatever the experiments we can perform in the BSL-3 facility, as well as getting the tissues out and doing immunohistochemistry and immunofluorescence studies. So that limited our ability in performing some Western blot or protein paradigms, but still we were able to get some good data from these studies. So as you can see, there is an increased AT1 receptor, significantly increased AT1 receptor expression in the amygdala of these acute SARS-CoV-2 infected mice. This clearly suggests that there is an increased inflammation, and then there is increased AT1 receptor, which is expected to be activated by renin-angiotensin system. So does this mice will have a changes in the B1 receptor expression? So here is the data both in the hippocampus and amygdala. So the B1 receptor is shown in red, and the neuronal marker NEU-N shown in green. So compared to the sham mice, the SARS-CoV-2 infected mice has a significantly increased B1 receptor immunoreactivity. So this clearly suggested that the acute severe COVID-19 disease induced in K18 humanized mice using the high dose of SARS-CoV-2 virus increased pro-inflammatory cytokines IL-6, TNF-alpha, and then the increased AT1 receptor. Not only that, but it also associated with increased B1 receptor expression. So this was until within the 10 days of severe disease model. So next, we wanted to extend the study to as much as possible. So what we did, we used the intermediary dose, and then these mice, as I mentioned, there was a 20% mortality, but the mice survived up to 45 days in this study. So we started doing behavioral analysis from the day 30, and then we euthanized all the mice at the day 45. As you can see, both male and female mice, their body weights are tracked until 10 days. There were no significant differences. However, we measured the SARS-CoV-2 antibody titers at the 45 days post-infection Even after 45 days, there was a significant increase in the SARS-CoV-2 antibody titers in these survived mice. Then we wanted to see the presence of virus. As you can see here in the lungs, compared to the mock treated animals, the SARS-CoV-2 treated mice, after 10 days post-infection, there is a significantly increased presence of the virus in the lungs. However, this virus was not present after 45 days. So that means the virus was cleared by 45 days. Then we looked at the brain, first the olfactory bulb, both two different kinds of antibodies. At the 10 days post-infection in this mild disease form, still the virus was present in the brain, but however in 45 days post-infection, this virus was cleared. And this virus presence was active replication virus. That means this virus was replicating in the olfactory bulb. So we wanted to see whether this virus can reach any other regions of the brain. So we looked at amygdala, prefrontal cortex, and hippocampus at 10 days post-infection. As you can see, there is a robust expression of virus, a robust presence of the virus in these brain regions, suggesting that the SARS-CoV-2 virus has neurotropism, and it can reach the deeper regions of the brain in 10 days post-infection. So one of the hallmarks of the SARS-CoV-2 infection is the lung fibrosis. So we wanted to see what happens in this mild disease form after 45 days post-infection. So we looked at both lung fibrosis as well as the B1R expression in the lungs. So here the Mason's trichrome staining we used to stain the collagen fibers in the lung tissue. As you can see, this is the various magnifications. So the collagen fibers are stained blue. Here is the quantifying data. There is a significant accumulation, a significant increase in the collagen fibers in SARS-CoV-2 infected mice. We also looked at the B1 receptor immunostaining, and this compared to the mark, there is a significantly increase. Here is the quantifying data. There is a significant increased expression of the B1 receptor in the lungs of this mild disease form. Interestingly, this increased B1 receptor expression was positively correlated with lung fibrosis. That means increase in lung fibrosis is associated with increased B1 receptor expression or vice versa. Then we looked at the neuroinflammatory markers. Similar to our acute study, so these, just remember, these studies were done after 45 days mice recovery from this virus infection. So even after 45 days, there is a significant increase in the amygdala, the IL-6, as well as TNF expression. So using real-time PCR, we also confirmed that in the hippocampus and prefrontal cortex, both IL-6 and TNF-alpha gene expression was significantly upregulated 45 days post-infection. So that means the mild SARS-CoV-2 virus infection, the increase in inflammation was persisted until even after 45 days. The mice were recovered normally. They were not showing any detrimental effect, but still there is an increased inflammation in the brain. Then we looked at the B1 receptor expression. As you can see, the three different regions of the brain, hippocampus, amygdala, and prefrontal cortex. And the neuron is the marker for the neurons. So the B1 receptor significantly, here is the quantification data. So in all three regions, there is a significant increase, both in the males and females. There is a significant increase in the B1 receptor expression. And this co-localized nicely with neurons as well as the microglia. So here is the IBA1, the marker of the microglia, and then the B1R in green. Here is the merge, is higher magnification, is suggesting that the B1 receptor expression is increased both in the neurons as well as microglia. Since B1 receptor is expressed in the microglia, we then looked at the microglial activation in these regions. So here, the quantification data of the area of the soma, as well as we did some other analyses like reduction in the number of branches, which is indicative of activated microglia, And we show that compared to the marked controls in SARS CoV-2 infected mice, there is an increased microglial activation, suggesting that microglia are activated and might be responsible for increased neuroinflammation. Um, these long COVID effects are mainly involved in some behavioral changes. So since we have the mice survived for more than 30 days, we started doing behavioral analysis from the uh, day 30 until 40, between the day 30 and the 45 days post-infection. So we looked at three behavioral tests. One is zero maze and Y maze and the open field. So zero maze is to look at the anxiety related behavior, and then the Y maze looking for like working memory and the exploratory behavior, and then the open field is for looking at the defects in the motor behavior. As you can see, in case of the zero maze, the heat maps showed that the mice spent less time in the open areas of the zero maze, suggesting that these mice developed some anxiety related behavior. So they have some defects in anxiety related behavior. In case of the Y maze, the arm entries or exploratory behavior was reduced in SARS-CoV-2 treated SARS-CoV-2 infected mice. So when we looked at the overall data in the open field, there were no significant differences, suggesting that their motor behavior was not drastically changed. However, they have some deficits in either in their working memory and exploratory behavior or some anxiety related behavior. We were able to do only these three behavioral tests because of difficulties associated with BSL-3, and we are interested in doing more behavioral related tests. Where hopefully we can do that in the future. So overall, our studies suggested that SARS-CoV-2 infection can increase B1R activation in both acute or severe disease states as well as a mild disease state, even until up to 45 days post-infection. And also, in case of the mild disease or the mice recovered from a mild SARS-CoV infection, there was an increased lung fibrosis, increased lung B1 receptor expression, there is an increased neuroinflammation, and then there is increased persistent neuropsychiatric symptoms, suggesting that B1 receptor might play a role in mediating these symptoms. However, so far I was shown that only the associations, so we did not do any mechanistic study. So in order to test whether blocking B1 receptor using a B1 receptor specific antagonist will protect the mice from the SARS-CoV-2 induced pathology. So in this case, what we did, we used the same intermediate dose of SARS-CoV-2 infection. And so I forgot to mention, so the virus strain what we are using is the original 2020 alpha variant, Washington alpha variant. So we wanted to, we infected the mice intranasally with this virus. At the same time, we used a B1 receptor specific antagonist, SSR 240612. So this is a non-peptide B1 receptor specific antagonist that can cross blood-brain barrier. So that means it can have protective effects even in the brain. So we used two routes, feasibility studies. So we used asthmatic pump to deliver the drug for 28 days, or we used an alternate day injection of higher dose. There was some technical difficulties in dissolving this drug and proper delivery. So we wanted to try both methods. So the idea is to do the behavioral test around the day 26 and then tissue collection at day 28. So we are still analyzing the data. The preliminary findings are very encouraging. We started with the lung fibrosis because that was one of the important finding. We found that there is an increased lung fibrosis which correlated with B1 receptor expression. As you can see here, the preliminary data suggests that compared to the mock or vehicle-treated mice, there is an increased collagen deposition in the lung, which reaffirmed our previous findings. However, when we treated with SR, which is the B1 receptor antagonist, it was able to reduce this fibrosis or collagen accumulation in the lung, suggesting that B1 receptor blockade will have some protective effects in preventing the lung fibrosis. We also have some preliminary findings from the behavioral analysis that these mice fared well in both zero maze and Y maze. So still we need to do a lot of these studies. We are working hard. However, as I mentioned, still it is going to take some time. So far, our data really, I hope that I can convince that the B1 receptor plays a role in increasing in neuroinflammation in both acute as well as a mild disease of SARS CoV-2, a mild disease of COVID-19. And we believe that this model we can use to further explore the long COVID effects, or at least the persistence effects of SARS CoV-2 infection and where the mice are recovered well. So still there are several questions. What is the best animal model to study the long COVID? So we were using the K18 human H2 mice, but there are some criticism from the scientific community that these mice are not natural hosts because in these mice the human H2 is required for the SARS-CoV-2 to infect the mice. So they are under keratin 18 promoter. So these are transgenic mice. So another alternative approach is to overexpress human H2 in the respiratory epithelium using intranasal adenovirus. Then that can be also used as a model, or we can use the naturally infected animal models such as hamsters or other models. Still, the debate is going on, which is the best model to study long COVID. We believe that the K18 human H2 mouse, our mouse model is very good because it can cause mild disease. At the same time, it is showing the neuroinflammation and neuroinvasion potential and some neurocognitive changes like behavioral changes. So we believe that our model is a good model to study the effects of the long COVID. And what is the best suitable virus strain? So we use the alpha variant of the SARS-CoV-2, but we are testing Omicron variant also right now. There are other mouse adapted SARS-CoV-2 viruses available. The advantage with this is that the normal or any other knockout mice or transgenic mouse, normal background other than the K8 human H2 can be infected with this variant. Contribution of the neuroinvasion potential. So there are conflicting reports that some studies showing that the virus in presence in the neurons or in the brain or in the several organs having some pathological changes. However, there are studies showing also that the virus is not reaching the brain still in certain human COVID-19 postmortem reports showing that the virus was not there in the brain, but still it has a severe disease phenotype. So still it is under investigation. One thing I was very much interested in looking at is the evidence for the B1 receptor involvement in long COVID using human biopsy or autopsy samples. Unfortunately, it was very difficult to get these samples. I'm looking for some collaborators or some direction. If someone knows anything, please let me know. That there is a recent study showed that using healthy as well as severe COVID infected plasma samples, the B1 receptor agonist, the desarginine and bradykinin levels were increased. So that is promising that our studies have some translational significance. Then the important question is how to differentiate the direct or indirect effects, whether the virus needs to reach various organs to cause pathology or the indirect effects through the cytokine storm. So still the debate is going on. And then the important question is, since we have shown some evidence that the SARS-CoV-2 infection can cause some behavioral changes, does SARS-CoV-2 infection increase the risk of development of Alzheimer's disease or Parkinson's disease in the future? Um, our studies show that there is some increased risk of behavioral changes, but still it needs to be explored further. So with that, I would like to thank my lab members and specifically to Dr. Eels because he's the one who is doing all the PSL3 work, and Dr. Shah Aplo, who is actually grown these viral strains, and Dr. Dorcas, who is the uh, head of the Department of Comparative Medicine. And with funding, we received a Brody Brothers Endowment Seed Grant to study this further differences between the Alpha variant and Omicron variant. And then we also, Dr. Eels was the PI on an American Parkinson's Disease Association initial grant to see how this long COVID model can be used to see the increased susceptibility to Parkinson's disease. With that, I will thank you everyone. And if you have any questions, I'm happy to answer. And thank you for this opportunity again.
Wonderful. Thank you, Srinivas, for that great presentation. Really covered a lot of stuff, and also, really appreciate the background behind this. It's actually really impressive that this managed to connect so many threads from your doctoral and postdoctoral work, and you've put together really a beautiful story here. If folks have questions, they can, raise their hand, put them in the chat, or just feel free to speak over me. I'll go ahead and get us started, though, with a question that I had. In determining the cellular source of some of these inflammatory cytokines in the brain, I'm guessing flow cytometry was infeasible because of the BSL-3 scenario. Were you unable to sort of, like, I kept thinking like do flow, but I'm guessing that was a BSL-3 issue. Is that right?
Yes.
Yeah.
That's the major. Even we were not able to take the plasma samples out. So we have to do all our ELISAs and everything in the BSL-3. So the only tissues so far we get out is the tissues which are stored in the formalin for more than 10 to 20 days, and then we were able to do. So that's why we had to rely on mainly on the immunohistochemistry, immunofluorescence studies.
Yeah.
But hopefully we are planning to do some ELISAs and some RNA gene expression studies in the future. Hopefully that will help us to tie together all the pathways.
Yeah, and obviously, the data you were showing were, sort of microglia focused, and as the resident immune cell of the brain, I think that, makes a lot of sense, and you're able to see some, inflammatory responses in those. I guess, are they just too sparse? Like if you had flow, or you were going to do, like in situ single cell sequencing and paraffin embedded, tissues, would you expect to be able to detect, like T cells in your model, and if so, like be able to investigate, if the brain infections drove increased T cell trafficking potentially?
Actually, yeah, we are planning to do some immune cell infiltration markers like CD3 and CD63, all CDs.
Yeah, right.
So the initial experiments, we would focus mainly on this inflammatory cytokines. So now we are trying to tease out what are the mechanisms. So especially we are interested in differentiating between the neuroinvasion effects as well as indirect effects. So we are planning to do some more immune cell infiltration markers in the near future.
Yeah, I think the the effects on the resident immune cells were really pretty striking. And so, yeah, then this question that, also arises in other sort of neurotropic viruses, like everything in the herpes family of, how much T cell trafficking to the CNS do they elicit? Or, there's this other cool piece about varicella, at least, is that it doesn't infect T cells that home to the skin and the brain. It's that once it infects T cells, it alters them such that they become brain and skin homing, right? And so there's some really interesting kind of neuroimmunology questions there that you might be really well suited to ask. And so, anyways, this is great work. I really appreciate you sharing it with us. I will stop talking and open it up to others who might have some questions.
Jump in. Is the B1R blocker something that's commercially available, or is that still a research only sort of drug? I'm just wondering what kind of drug it would be.
So these B1 receptor antagonists are all not as a drugs available, but they are available from the companies. So there are several different both peptide and non-peptide antagonists. So there are small molecules too. So there are several. We were focused on two of them, like R715 and SSR, because R715 will not cross blood-brain barrier. Then the SSR crosses blood-brain barrier. So that helps us to delineate central versus peripheral effects. So both in cardiovascular diseases as well as in the SARS CoV-2 model. So yeah, they are available, but not as a drugs. Just
Gotcha. This is fascinating. Thank you.
Thank you.
For the behavioral experiments, I guess I'm wondering, is because I was a little unclear, I missed part of the thing on how the setup was, but with those mice then who have had sort of long COVID and have COVID in the CNS, they would also ostensibly have had like lung fibrosis and whatnot, right? So like you can't, as of yet, sort of tease out if some of the behavioral effects are due to COVID in the brain versus COVID in the lungs, and they're just, less active in certain ways. Or were you able to sort of separate those effects?
And we did not like tease out like each mouse, whether it has changes or whether it has fibrosis, but most of the like our end size is reasonable, from 5 to 10 mice in each group, and we were able to show that the significant effects in at least cohorts of mice which we tested for the lung fibrosis after 45 days post-infection. And also all the mice, there is a significant effects. There is a average significant effect in behavioral changes. So unfortunately, we were not able to tease out. So in order to see the brain infection, so this virus was cleared after 10 days. So we were able to see during the 10 days post-infection. So after 45 days, I cannot say whether it reached the brain and caused the changes or due to some other effects. We need a marker which can show that, okay, these brains are infected with virus. But unfortunately, we don't have any specific marker to say that.
Right. Okay, that makes sense. Yeah, I was just sort of thinking about the neurobehavioral changes and, whether or not that's able to be sort of assigned to specific, or to organ specific differences. I'm thinking about like last time I had COVID, and like, I don't think I'd have wandered out into any big rooms or parks either, but hopefully that wasn't because my neurologic function was altered, and rather just because my breathing was crummy. So okay, it's good. I appreciate that.
There is lots of unknown. This is unknown territory. Like every day there are new papers coming up with new scary things. So it takes some few years to clearly understand whether it's good or bad. We don't know, but there are some changes definitely going on in these mice. So that's what we are trying to understand.
And I guess if you had unlimited funds and personnel, right, like that magic scenario, what are there additional COVID strains that you'd want to look at? And if you had to pick like the next one after sort of COVID alpha, like would you be going to Omicron, to whatever's most temporally relevant? Like how would you sort of pick that out to think about what other strains to investigate?
Yeah, so that's what we started. We started with alpha variant because that's what available at that time. But when you just see the distribution, like the people infected with which variant most, so most probably that will be the Delta and the Omicron.
Yep.
So if I have funds, I wanted to compare the Delta and Omicron compared to the original alpha variant. So that's what we proposed, and we were right now testing the Omicron variant. But the one major difference between the alpha variant and the other variant or the Omicron is mild disease. So we need to again start from scratch, defining which it can cause mortality in mice or severe disease in mice, and then which dose will actually have effect.
Yeah.
So,
well, and if they would reclassify any of the extant strains like Omicron potentially as being BSL2, that might motivate you to work with one strain more than others potentially as well. So, and I think that's going to eventually happen, but it'll take time, and regulatory stuff is, as you know, slow to move and adapt. So
that's true.
This is awesome. Anyone else? We have maybe a minute left for one more. Heather, yeah.
Yeah, so I was going to ask, if this were going to translate to humans down the line, any just thoughts, like if you had to take your best guess as to what the side effects or unintended consequences of manipulating, this particular receptor, small molecule, because there's always some unintended, undesirable effect. So what would you think would be the biggest downside if you were trying to get a therapeutic effect based on
that's a good
based on this small molecule?
Yeah, that's a very good question. So the one important thing about the B1 receptor is normally not expressed. It's only expressed under stress conditions or injury, tissue injury, or inflammatory conditions. So there is a B1 receptor whole body knockout mouse model in my lab, and these mice are fertile, survive. They don't show any gross abnormalities. That means if you knock down this receptor even at the birth, it's not going to have any disease phenotype at the baseline. And these mice are protected from hypertension, angiotensin 2 induced or dopamine induced hypertension. So far, I didn't see any side effects in mice, but I'm not sure about the humans. But it's a promising results what we have. So since the knockout mice did not show any significant disease phenotype, I believe that it's not going to have that many serious side effects.
Yeah, no, that would be very reassuring, obviously, to partners and investors. I would still worry that it may not have any phenotype in mice at baseline, but if it appears during stress,
yes,
so it may play some sort of a role during stress that we would be sad if it weren't there. But
we don't know. Unfortunately, it is not any clinical trials right now. So maybe in the future, it will go into clinical trials.
Time will tell. Yeah.
That's true.
All right. Well, thank you, everybody. We are at time. I appreciate everyone's attention. And Srinivas, this was just a wonderful, really fascinating presentation. And I think answers several important questions and raises many more, which is always my favorite kind of talk. So thank you. Thank you for participating, for your time today. Appreciate it.
Thank you so much. And thank you for the opportunity. I'm looking forward to involve more in this ADRC and ADRT.
Thank you.
I'm looking
Great
forward.
job. Thanks.
Thank you.